Hello students, in this video you will learn about the process of mitosis. Basically, mitosis is the process of cell division in which one cell gives rise to two genetically identical daughter cells, resulting in cell duplication and reproduction. The number of chromosomes is preserved in both the daughter cells. Mitosis is divided into two phases. Number A, karyokinesis, and number B, cytokinesis. Karyokinesis is a nuclear division in which nucleus undergoes a series of changes to form two daughter nuclei. Karyokinesis is further divided into interphase, prophase, metaphase, anaphase, and telophase. Number 1. Interphase. Interphase is a part of the cell cycle where the cell copies its DNA as preparation for the mitotic phase. In interphase, metabolism of the cell increases and it is often termed the most active phase of the cell cycle. A series of metabolic changes occur during this phase, all of which are divided into three subgroups, which is G1 phase or pre-DNA synthesis phase. It is the longest phase of the cell cycle and is followed by the mitotic phase of the previous cell cycle. It is also termed as the resting phase as no DNA synthesis takes place during this phase. However, during G1 phase, several organelles increase in size and cell rapidly synthesize different types of RNA and proteins. S phase involves the replication of nuclear DNA and the synthesis of histone proteins. The replication of cytoplasmic DNA can take place at any phase in the cell cycle. Thus, at the end of the S phase, each chromosome has two DNA molecules and a duplicate set of genes. G2 phase is termed the second gap phase or resting phase of the interphase. During this phase, the synthesis of RNA and proteins required for the cells continues. Cell division involves the enormous expenditures of energy, thus cell stores ATP in the G2. By the end of this phase, the cell enters the division or mitotic phase of the cell cycle. Prophase. Prophase marks the beginning of cell division and is the longest phase of mitosis. During prophase, chromatin network undergoes condensation and forms definite number of chromosomes. Prophase is further subdivided into three phases, which is early phase, mid phase, and late phase. Early phase, DNA molecules condensed and form short chromosomes. In this stage, chromosomes appears like a ball of wool, so-called spireme stage. In animal cells, centriosomes, or called centriole, divided into two and moves towards opposite poles. Centrioles produce out fine microtubular fibers called ester trace. Plant cells do not contain centrioles, so-called acentric mitosis. In mid-phase, chromosomes shift towards the periphery and leaves a clear area. It becomes shorter and thicker. Each chromosome consists of two longitudinal threads called chromatids. In late phase, spindle fibers starts appearing around the nucleus and size of chromosome is much reduced. Spindle poles are formed without esters in plant, with esters in animals. Prometaphase. Prometaphase is initiated with the breakdown of the nuclear envelope, which enables the interaction of spindle fibers with the chromosomes. At this stage, the chromosomes are violently rotated and oscillated back and forth between the spindle poles because their centromeres are capturing the end of microtubules and are being pulled by the captured microtubules. By the end of prometaphase, the sister chromatids are attached to the spindle fibers on the opposite ends and are held on the metaphase plate. Metaphase. During metaphase, the chromosomes are shortest and thickest. Their centromeres of the sister chromatids occupy the plane of the equator, forming a metaphase plate, and the arms remain directed towards the poles. Two chromatids of a chromosome repulse each other with the microtubules remaining stationary and under tension. Anaphase is the shortest stage of mitosis. The centromeres of chromosomes start divide into two. Two daughter chromatids are separated and daughter chromatids are migrate towards opposite poles. Spindle fibers are attached to the centromere and pull the chromosomes to the poles. As their kinetochore microtubules shorten because these microtubules are attached at the centromere region. The chromosomes move centromeres first. The cell elongates as the non-kinetochore microtubules lengthen. By the end of anaphase, two groups of chromosomes are formed, one at each pole. Telophase. The end of the migration of the daughter chromosomes to the poles marks the beginning of the telophase. During telophase, the events of prophase occurs in reverse sequence. A nuclear envelope reassembles around each group of chromosomes to form two daughter nuclei. Events like the disappearance of mitotic apparatus, reduction in the viscosity of cytoplasm, followed by the synthesis of RNA take place during telophase. The central chromosomes resume their long, slender, extended form and the nucleolus appears at the end of telophase. Now, cytokinesis. Cytokinesis is the division of cytoplasm which is followed by mitosis, resulting in the formation of two separate daughter cells. Cytokinesis usually begins in anaphase and continues through telophase and into interphase. In animals, cytokinesis occurs through constriction and furrow formation. The first sign of cleavage in animal cells is constriction of plasma membrane during anaphase. In plants, however, cytokinesis occurs by cell plate formation as constriction is not possible due to the presence of a rigid cell wall.